If you have matched paired DNA-seq data between tumor and normal from the same subject, you can run the var-scan2 matched pair analysis. This annotates somatic mutations, that is, it compares two samples from the same subject, such as tumor or normal, for differences in genotype. It will generate calls for the genotype of the samples from the subject and flag germline versus somatic mutations. This module requires additional metadata columns in the design table, including a subject grouping column and a tumor versus normal grouping column. You can also include additional metadata columns as well. In this design table, the subject ID can group between tumor and normal, and the column tumor or normal indicates the tumor status. To run the module, click NGS, Variation, Summarize Matched Pair Variant Data. Choose the project and the data, which references and observations to use. Choose the pairing column, subject ID, and tumor status column, tumor or normal, and choose the status that indicates normal samples. Specify whether to annotate by dbSNP and whether to generate a merged or individual VCF files. Because Array Studio is moving toward a VCF-based analysis, we recommend outputting a merged VCF report. Under Advanced, you have additional control over pile-up and variation options, as well as filtering options. After you run your analysis, you can choose to either directly view the mutation report, or you can annotate your mutation report or your VCF file with online annotators. Under NGS, Variation, Annotate Variant Table Report, or Annotate Variant Files. We recommend annotating a merged VCF file, which will generate an OSCR, or Omicsoft SNP classification result object. This data object is sufficient for filtering and sorting of your data. For each variant along the genome, the reference and alternative allele will be indicated, as well as the distribution of genotypes among your samples. Clicking on a row will give you additional details about the samples and the genotypes. Based on the gene model, you will also find predicted changes, including the location in the gene model, the change to the protein, and information from the selected online annotations, such as 1000 genomes, frequency, and clinical significance. You can filter by gene name or by functional annotation. When you look in the Details window, you will find a column that states the somatic status of the variant. Right-clicking on a row ID will allow you to open a new Genome Browser window. you will see your NGS data coverage for the region, and you can show variation, and you can also show an alignment profile. Here you can see the sequence changes directly located on the individual reads